Hello and welcome everyone. In this podcast, we're going to compare bacterial chromosomes and their organization and their overall structures to that of chromosomes found in eukaryotic cells. So let me draw a bacterial chromosome. In general, bacterial chromosomes are circular. You will find some examples where the bacterial chromosome is linear, but as a general rule, these chromosomes will be circular. So if asked on a test or a quiz, you would say they are circular. They will have one origin of replication. So I'll write that over here as well. One origin of replication. And I'm going to abbreviate replication like so. As a general rule, though there are exceptions to this, bacteria will have one type of chromosome. As we saw in the previous slide, when we were talking about eukaryotic chromosomes, we made note that there were many kinds of types of chromosomes. In humans, there are 22 pairs of chromosomes. In bacteria, there's usually just one type. However, there can be a single chromosome or multiple. So we talked about how in eukaryotic cells, the chromosomes were either haploid, present in one copy per chromosome, or diploid, present in two copies. Bacteria can be a single copy, or they can be 2, 3, or 4 or more. We would say that a chromosome, usually in bacteria, is a few million base pairs. Seems like a lot, but when we start looking at eukaryotic chromosomes, we'll see that this is very small. And it's small because bacteria are very efficient organisms. Probably the most efficient organisms that we know of. They're also quite versatile. They can live in a variety of types of environments, and they can adapt to those environments much quicker than a eukaryote could. The bacterial chromosome is very streamlined. There's not a lot of extra spaces in the chromosome. And what I mean by that is that in red here I'm going to draw genes. So here's a gene, here's a gene, here's a gene, here's one. I'm going to see a pattern here pretty soon. You can see there's very little space in between the genes. So over here, let's write genes tightly packed. Maybe not the best way to say it, but it gets the point across. That we don't have a lot of space here. Or we could say little intergenic space. And that would be these little spaces in between the genes. We'll see that in eukaryotic cells, much of our chromosomes are made up of what we call repetitive sequences. And that's why I left some of these larger spaces here. In bacteria, there's not a lot. There is some, but there's not a lot. Whereas in eukaryotes, most of our chromosomes are these repetitive sequences. So I'm going to put a few repetitive sequences. They don't code for genes, but they have important functions. And I don't have room to write it here, so I'm going to write it over here. So these repetitive sequences are important in gene regulation, and DNA folding when we package the chromosomes. Okay, I want to erase this now and spend some time talking about how this DNA is folded. So in thinking about how bacterial chromosomes are folded, I want to first draw a bacterial cell. So when we look in a bacterial cell, typically what we'll find is a single structure. Sometimes we might find two, sometimes more, but usually one or two. And these structures are called nucleoids. And what this structure is the folded up DNA with proteins. And it's the proteins that help in the folding of it, as well as those repetitive pieces of DNA that I mentioned on the last image. They're so tightly compacted that if you were to lyse this cell by poking a hole in it, what would happen is the DNA within this cell would explode. Well, explode is probably not a good word. It would unravel rapidly. So here's our cell that's exploded. The DNA would rapidly come out of the cell. I'm trying to exaggerate here a little bit, but it's really not exaggerate much, because what's compacted in there is many times the size of the bacterial cell, which illustrates in this drawing here. You can see an actual picture of this in your textbook, but it demonstrates to you here how tightly the DNA is compacted with these proteins. So you can imagine it would be really hard to just cram this in there unless it was structured and compacted in a certain way. I think of this exploding chromosomes here out of the cell kind of like if you had those cans you can buy and there's these spring snakes in them, and when you open it up, this snake shoots out. That's what I think of is happening here. I also think of the way that the chromosomes are being packed is the way you would pack a suitcase if you're going to go on a vacation. Maybe some of the first times you packed a suitcase, you just crammed a bunch of stuff in that suitcase. And when you got it out, your clothes in it weren't very useful because they were all wrinkled and you couldn't pack as much in there. Then you might have learned that if you take your clothes and you roll it up nice and tightly, your shirts and your pants, roll them up really tightly and put them in the suitcase, you can get more clothes in your suitcase, and then when you get to your destination, your clothes aren't as wrinkled and they're in a more usable, ready-to-use fashion. That's how DNA is packaged. It's packaged in a very organized way so that you can get all this DNA here stuffed into this tiny cell. And not only is it stuffed in that cell, but it's in the cell in a usable way. It looks like just a big ball of yarn here, but in reality it's structured in such a way that enzymes can find the DNA necessary to turn on genes or to copy the chromosomes. So let's talk a little bit about how bacterial chromosomes are packaged. So let's talk about how we're going to get this rather large chromosome packaged into a single cell. So I'm going to draw the circular chromosome around the borders of the entire board here. So this is our circular piece of DNA from our bacteria. And we need to get this inside of a cell, and my modeling is off a little bit, but say a cell that is about this big. In reality, the cell would be much smaller than that, but you wouldn't be able to see it if I made it that small. So the first thing that it does is it takes this chromosome that I have around the edges of the whiteboard here, and it begins to pinch them in to the center in such a way that it looks like this. And I'm going to draw this structure here, which are DNA binding proteins. I'm going to just write that down here so we remember that's what they're called. DNA binding proteins. A fairly clever name for a protein, since that's what it does. It binds DNA. And as it's binding the DNA, it's going to pull it into the center. So you get something that looks like this. And as we can see here, what's happening is this very long piece of chromosome now has taken on a much smaller arrangement. So again, let's draw our arrow to our binding proteins. And these loop structures here of the bacterial chromosome, these are called loop domains. Now what happens is, and this will be much more difficult for me to draw, But these loop domains now begin to twist upon themselves. And so I'm going to attempt to draw this, like so, where they just begin to twist on each other. And we call this process supercoiling. And because I can't draw molecular structures exactly the way they would be, this isn't going to fit right in there. But the way the bacteria would do this, they would do it in such a way that it would compact this large piece of circular DNA here by first forming these loop domains and then supercoiling it to the point where it would occupy a very small spot in this bacterial cell. It's a fairly efficient process and it works quite well. And again, as I said, this supercoiled structure here is designed so that it packs into the small cell, but it's also usable. Okay, now that ends this podcast, and I want to just summarize real quickly what we talked about. We talked about the structure of bacterial chromosomes. We talked about parts of them. We talked about their efficient size. We talked about the spacing of the genes and the repetitive DNA sequences. We talked about the nucleoid, what it was made of, and the space it occupied inside of the bacterium. And then lastly, we talked about how the bacterial chromosome was packed. So it was a packing of the bacterial chromosome. Again, using those loop domains and the DNA binding proteins and the supercoiling to get this rather large chromosome packed efficiently into the bacterial cell. In the next podcast, we're going to talk about the structure of eukaryotic chromosomes and how they pack their chromosomes inside of a tiny cell. If you have any questions, please let me know, and I'll see you in class. Bye.